Thank you for the introduction. My background is in detection of mosaic chromosome alteration. That's kind of what led me to do some work on the field of non-invasive prenatal testing and IPT. So we'll talk about how chromosome alteration affect pregnancies and some other disorders that people care about. If I think about clinical care for pregnancy, what kind of things do pregnant mothers in clinician want to know about a pregnancy? There's a long list. These are the kind of things that I'm going to touch today, which are on a first order approximation, the things that physicians care the most about. Autosomal trisomies, microdeletions, sex chromosome trisomies, recessive mutations, and if we have time, we'll also touch a little bit about de novo mutations. We will talk a lot today about sensitivity and the positive predictive value of PPV of a screening test. Just as a quick reminder of what these are, the sensitivity is the detection rate. Like if we have a disease in a working population and we run tests on everybody, like what percentage of individual with that disease or that type of mutations do we find? And the positive predictive value is the amount of individuals that receive a positive result that actually have that condition or that mutation. So a good screening test is going to be something that has high sensitivity and a high PPV. And as a side note, it's important to understand that PPV is something that is kind of easy to measure. Once you design a screening test, you just follow up with everybody that got the positive results and you check whether that was really the case. Sensitivity is a difficult measure to assess because you really have to follow up with everybody. You really have to figure out something like a ground truth or like who has a given condition and then see how your screening test performs. And so you need clinical trials to do that. And it's especially difficult for conditions that are very rare, so that if you want to assess the efficacy of the PPV, you need to find, so the sensitivity, you need to find a lot of cases in your cohort. So I'll structure the talk in two parts, one dedicated to chromosome alteration, one dedicated about single gene disorders. In the interest of time, I'll talk about chromosome alteration first, and if there is enough time, I'll discuss the second. So what are we looking for with respect to chromosome alteration in a pregnancy? Turns out that that question, it's very important to understand this, it's different whether you think about it, what you can find at the beginning of the pregnancy or what you can find at the end, in the newborn. The reason being that there's a lot of natural selection going on already at the stage of the pregnancies. There is a lot of things that can go wrong from the very beginning that are just not viable, and so that you'll never find them in newborns. But so, for example, if you look at around 10 weeks gestation age, so towards the end of the first trimester, you'll find, for example, a lot of, here in yellow, called other autosomal Ts, trisomies beyond the common ones were trisomy 21, trisomy 18, and trisomy 13. As you progress in the pregnancy, you don't find those anymore. They end up in fetal demise, miscarriages, and then they don't get found anymore in the second trimester and then newborns. By the end of the pregnancy, you only find the things that are viable, right? Or you can say are compatible with life. And so you find a lot of, when you restrict to newborns with chromosome alteration, you find a lot of trisomy 21, trisomy 13, trisomy 18. SCA stands here for sex chromosome alteration, some other things. And so the value for a screening test for chromosome alteration lies a lot in being able to find these trisomy that are viable. You could find interesting to also be able to detect the other ones, but those will end up in the pregnancy and terminate itself anyway, so they won't have as much clinical value. And this is maybe a fun fact. Why are trisomy 21, 18, and 13 the ones that are viable that we are interested in? You probably have heard that these are the small chromosomes, and you would be right in that, but they're not the smallest. It's actually what defines even better what's special about 21, 18, and 13 is that they are the ones with the smallest number of genes in the human genome. I also, if you don't believe me, you can run these 2 lines of code and get the same numbers. But turns out, yes, trisomy 21 is absolutely the one, the smallest number of genes, and the 18 and 20 and 13 have more. And they turn out to be the viable ones. With viable, though, as a caveat, I think everybody has heard of trisomy 21 Down syndrome. Maybe not everybody has heard of trisomy 18 and 13 before. And that would be because trisomy 18 and 13, although they can progress up to the birth, they have an extremely short median survival. Most kids die in the first days of life. As a result, clinical care for these kids is usually palliative. You just wait until the demise. While trisomy 21, it's a condition that with the advanced medical care these days actually has allowed children born with an extra chromosome 21 to actually survive into adulthood and to have a life expectancy of 50 to 60 years. So shorter than fully euploid humans, but still very high. And so trisomy 21 was actually, exactly because children make it into adulthood, was actually something known for a long time. It was first classified as a syndrome in 1862 by John Langdon Down. And in reality, at the time, it wasn't even referred to as Down syndrome. It was referred to as Mongolism, and people with Down syndrome were referred as Mongoloids. And that's a little bit of a history behind there. It's an unfortunate terminology that made it until the end of the 20th century. For those like my age or older, you probably must have heard it as you grow up. And maybe the young ones in the crowd, maybe you never heard of it. And so even if it was known since the 19th century that there was a syndrome with a certain prevalence, and it was not known the genetic cause until 1959. In the 1950s, there was a time when lots of new discoveries about DNA happened. 1953, the DNA helix was discovered. And in the 50s, some new methodologies to culture cells were developed that allowed scientists to culture cells and then with microscope to photograph the chromosome and count them for the first time. That caused a lot of activity back then. And in 1958, a scientist in Paris, Jérôme Lejeune, identified for the first time a case of an extra chromosome in a child with Down syndrome. Then the same year, more cases, he identified more cases. And in January 1959, for the first time, he published a note in a journal where he mentioned that 3 different kids, they preferred as Mongoloids at the time, show the presence of 46 instead of 46 chromosomes. And that's when clinicians started to realize, wait, so maybe this is actually what's going on. There is an extra chromosome, that's what's causing the syndrome. For actually trisomy 18 and trisomy 13, because they're not as viable as trisomy 21, and because also they are less common, there wasn't a name for it until 1916. The discovery of this syndrome actually came with the discovery of the genetic cause. And very interesting, in 1960, 2 manuscripts were published in the same issue of Lancet. One by John Hilton Edwards that described an individual with trisomy 18 that didn't look like a Down syndrome kid. And he identified an extra small chromosome. He said that it was an extra chromosome 17. Turned out actually that was an extra chromosome 18 and took like a year to figure out the mishap. But the discovery stand and actually the name stood as well. And now we refer as trisomy 18 also as Edwards syndrome. And so in the same issue, then another paper by Klaus Patau identified another kid with a syndromic, but not with Down syndrome features, that also under analysis of culture cells showed that there was an extra chromosome. And I find also this very interesting. Here in this case, it was clear that there was an extra chromosome of the type D, but given the limitation of technology back then, it could not tell whether it was 13, 14, or 15. These are this large acrocentric chromosomes that look a little bit alike under the microscope. And it turned out later on that it was an extra chromosome 13, and then trisomy 13 became also known as Patau syndrome as a result. And so if we ask about this autosomal trisomy and other chromosomal alteration that can make it into birth, what's the prevalence of this event? One way to think about it is to think about it in the terms of out of 100,000 newborns, how many will be affected? And if you think about in these terms, maybe something like 140 will be affected by trisomy 21, 13, or 18. Mostly it's trisomy 21 that is the most prevalent. There is also other things that we can test nowadays, like microdeletions, which they are less common in older mothers, but actually they're even more common in younger mothers. This is because of the fact that autosomal trisomies grow exponentially in prevalence as the mother becomes older. While microdeletions, they happen through a different genetic mechanism, so actually the prevalence stays the same. And you can see this graph where, again, for younger mothers, Down syndrome is something very uncommon, but when you get to the age 40, Down syndrome is something that happens with a prevalence of more than 1% of viable pregnancies. And so there's been a lot of interest in trying to screen for these in the beginning, especially for mothers older than 35. Already back in the 80s, there started to be a development of what we call maternal serum screening test for autosomal trisomies. The idea is that when an autosomal trisomy is present in the fetus, some aspects of the fetus will be, and the placenta will be dysregulated. And this will cause the prevalence of hormones and proteins in the maternal serum to be different from what you would observe in a fully euploid pregnancy. However, these measurements are very non-specific. There are differences, but it's not deterministic. And so you can statistically see a difference, but you don't get a very good predictor out of it. Over the years, multiple different measurements got combined. I'm not going to go through what each one of it is, but the AFP, HCGH, S3, DNA. Eventually, they came together to make what it's called the quad test that used to be performed maybe in the 90s, where everything gets measured maybe at the end of the first trimester. There are additional ultrasound measures that get made, and altogether, those standard tests that used to be done allowed to devise a screening test that was not very specific, but still had a 90% sensitivity for Down syndrome, but it still had a 3% false positive rate, which doesn't sound much, but it means that 1 in 30 pregnancies back then would basically get a high risk result, saying like, you might have something. And then when you went to look at those pregnancies, only maybe 1 in 18 would actually have something, again, because the test was very not specific. The positive predictive value was very low, less than 10%. And that basically meant that all those women with high risk results had to undergo amniocentesis to confirm diagnostically whether there was something or not. And the majority of those amniocentesis will just return all good. Also, there is something to say about the fact that the amniocentesis carries a little bit of risk. In the literature, they say 1% miscarriage rate. Though that's a complicated thing to measure. And so lots of parents, arguably rightly so, they will also sometimes say, like, I don't think this is specific enough, I don't want to do it, and I'll just carry on with the pregnancy without. And so amniocentesis, it's actually, or these maternal screening tests, they're not diagnostic because, again, the positive predictive value is so low. They always was recommended to follow up with an amniocentesis to confirm the presence and to provide a diagnosis. And amniocentesis was available since 1967. Technology is similar to what was used to discover actually the trisomies to begin with. And this is a depiction of what it looks like. You actually have to extract some amniotic fluid from the fetus. And so for a long time, or I'd say maybe like for a couple of decades, there was this situation where maternal serum screening tests were very imprecise, and there was this unmet need for a test that would be more accurate and non-invasive at the same time. And in 1997, actually was discovered for the first time that if you separate the plasma from the nucleic, the nuclei in the blood, and you look at the DNA circulating free in the blood, in the serum, you actually find that some percentage of that comes from the placenta of the fetus. the way they discovered this, they try to amplify the Y chromosome, the DNA in a pregnant mother with a male fetus. You shouldn't find any Y chromosome DNA in the blood, but actually you can see that they could amplify, and they could only do so in pregnant women with male fetuses. So that discovery gave the tantalizing understanding that there should be information in the blood to make a determination about what the DNA of the fetus should look like, or at least I should say the DNA of the placenta. There is then a little bit of discussion to be there to say about the difference between placenta and fetus that we won't touch. But the reality is that there is information there. But 1997, this was before the Human Genome Project was completed, so that realization didn't exactly translate into something of clinical relevance. That actually took a lot of years. And the first actually studies that show that it's possible, maybe at scale, to identify autosomal trisomy from cell-free DNA in the maternal serum came in 2008. They showed that by the randomly sequencing DNA from cell-free DNA from the serum, with shotgun sequencing, we could actually get enough molecules from the chromosomes 21, 18, and 13 to see that whether there was maybe an overabundance of molecules from the chromosome that could be indicative of trisomy in the fetus. And although, as you can see from these plots, if you look carefully, they weren't very precise. Some of these chromosomes, as you can see, they're extremely noisy. For trisomy 18, 13, and 18, you can see that there is separation between cases and controls, but it's not used separation. And here, there's another plot from other people. There were two papers that came out in PNAS in 2008. You can see that you're really looking for very, very small deviation in abundance. And the reason for that is duplex. Only about 10% of the DNA in this plasma comes from the placenta. The rest still comes from the mother. The majority comes from the mother. This is DNA that mostly comes from cells that die around the body. And then also, and this is kind of almost by evolutionary design, the chromosomes that we're interested in are the small ones because they're the ones that are viable. And so only a very small percentage of the DNA from the genome will be informative for those chromosomes. And so the summary is that we need to sequence a lot to see these deviations. And this was in 2008, before the Fast Genome Project. Whole genome sequencing was still very expensive. And so at that point, we could see like there was a path to do this, but the technology just wasn't cheap enough, which we all know then that changed very quickly. And so as soon as that happened, in 2012, there was a flurry of activity in the startup world. 4 different companies jumped into the space with their own non-invasive prenatal testing, where basically they kind of did what I just mentioned. They just separate the serum from the blood and sequence a lot of molecules and then look for deviation. There's a little bit of something to say about the fact that not all methods are the same. Some of these companies like Sequenom and Illumina, they just did low pass whole genome sequencing, still very expensive at the time. Some companies did some more targeted approach. Then there is this last company does something that it's actually qualitatively very different, which instead of massively parallel sequencing, shotgun sequencing, everything, they target the chromosomes of interest, in part because, as I said, they only a small percentage of the DNA would come from those otherwise, in part also because by targeting this thing called SNPs, they allow to use a method where instead of quantifying in absolute count, they quantify DNA in relative count, and that can be robust with respect to some biological noise. I won't say more to that, but just not all the NIPTs are the same, and I think this a lot gets very much lost in the literature. And the very first generation of an IPT, and these are studies from 2014 and 2015. So even if I'll say a bit more later, the latest generation is better than the first generation, were already outstandingly better than the standard of care provided up to then. So here you can see in the column standard screening, this is what the maternal seminal screenings of the 80s and 90s were like. Based on hormones and proteins in the serum. The positive predictive values, as I said, was less than 10% for trisomy 21 for those tests. The positive predictive value for this newest test is close to maybe here 50%. 90% for this other type of test. So even the first generation had an incredibly higher precision than maternal serum screening test. And this basically started a revolution in the standard of care for pregnancies. And what that also qualitatively meant in the clinic is that while before you would have a woman come for the first trimester screenings, if she had received positive results, you had to counsel, say like, we should probably do amniocentesis, but most likely it's going to be just a scare. Now the standard of care is like, it's not guaranteed. This is not a diagnostic test. It's always a screening test. But the majority of people that receive a positive screening test will also receive a positive diagnosis. So that changed a lot the conversation in the clinic. And also what changed a lot is that, well, suddenly now all these women that were receiving positive results before, now they didn't anymore. And so the rate of amniocentesis decreased 95%. Well, that was something that was routinely done in the clinic. Now it's done sporadically, basically. But it's still though done because, again, like there are cases where even this NIPT screening test turn out positive, but then it's just a scare. Because one thing I heard once that I like to repeat is that in biology, nothing is 100%. There is always those weird cases, you know, maybe the placenta and the fetus were discordant. And so you can never make a final determination from an NIPT, although it's a much better determination than we could do before. And then there's also another aspect that in the beginning wasn't talked much, that the sensitivity and the PPV of this test was provided as like a one number. It's also different it's difficult to correctly measure it when it's so high because you have so few cases in these trials to measure it. But the reality is that the sensitivity and the PPV, they're not the same for all pregnancies, as they are actually very dependent on much of the DNA that was assayed came from the fetus. And not all pregnancies will have this like 10% of the DNA coming out from the fetus. Some will have more, some will have less. It's very dependent on gestation age. In practice, what happens is that after implantation at 3 weeks, the placenta grows really, really fast up to 10 weeks. And then kind of stabilizes a little bit. And as it grows, the fetal fraction, the fetal DNA fraction in the serum also grows up exponentially. So you can't really do this test before 9-10 weeks gestation age, isn't enough fetal DNA. But even then, you'll still have some mothers that will have a very small amount. And then the sensitivity in the PPV for those cases is actually lower than what's reported in these trials. And this is actually, it's also a little bit of an issue because it will lead to what we call no-call rates. Sometimes the test will just return saying like, there wasn't enough fetal DNA, sorry. Either try again or seek another method for diagnosis. But it also, yeah, as I said here, it means that when the fetal fraction is low, it's going to be easier for the test to make a wrong determination of trisomic status. And this is compounded with this unfortunate phenomenon that actually some of the pregnancies that are trisomic, especially trisomy 13, 18, and maternal triploidy, happen to be more often the ones that have low fetal DNA fraction. Part of the reason that the trisomy comes with a lot of dysregulation, sometimes placenta, for example, is smaller, and smaller placenta then leads to low fetal DNA fraction. And so in a way, the thing you need the most to make a correct determination is the thing that happens more often when that correct determination is needed. So you have this weird interplay. And eventually in 2016, the American College of Medical Genetics actually said that this test should, or recommended that this test should always report the fetal fraction in the result so that the clinician could make even a better determination whether the test is reliable or not. And then, so beyond these aspects, after the first round of the first generation of an IP test, they started to add more features. One interesting feature that has been added in the last decade was that microdeletions are now tested. Now, each company does this in a different way. But if you remember the plot I showed before, 22q11 is kind of like the microdeletion that is the most prevalent across all microdeletions that you might want to test about. It's very prevalent because it's caused by errors in meiosis due to homology between regions of chromosome 22. And there was a lot of improvement over the years on this. The problem with 22q11 is that very small segment. It's like 10 times smaller than chromosome 21 if you want. So like only 0.1% of the DNA comes from that region. So if you want to determine that region, you really need to be careful about how you do the test. But the latest version of the test claims to have a sensitivity above 80% and a positive predictive value higher than 50%, which is amazing to think about this being possible. 22q11 wasn't even something that was available in the 80s and 90s, something you could screen. You might see something in the ultrasound, but there was no specific screening for it. And again, it's something that is more common than trisomy is for very young mothers. And then there is sex chromosome trisomy. This is something that was not tested. Sex chromosome trisomies are all viable, and actually they're not really associated with strong syndromes. XXY, maybe XXY was the only one that was actually classified as a syndrome before the genetic discovery in 1942. The other ones, XYY and XXX, were only identified from genetic studies. And the reason is that most people that have sex chromosome trisomies, they are unaware that they even have it. The rate is a bit higher than autosomal trisomies. However, they don't grow in prevalence with age as much. And they're not all 90% of maternal origin like they are autosomal trisomy. In particular, XYY can only happen from the father. There is no doubt about that. We now actually know that, although again, most people don't know that they have them, if they have them, because the phenotypes are not very specific. The only kind of exception there is XXY individuals. A lot of them turn out to be infertile, and so when they try to have kids and they seek medical care, then sometimes they discover that they have an extra chromosome X. Other than that, the rest they often don't know. And we didn't really know the phenotypic spectrum of these syndromes before, because we could only ascertain the individuals that somehow, for some reasons, had received the diagnosis. The first study actually that looked at this in an unbiased way came out only a couple of years ago, using the data from the UK Biobank. And it showed some interesting things that, well, this is known, XXY and XY individuals are taller than euploid males, but also show that the rate of childlessness is higher in XXY and XYY individuals, and this is not just for infertility reason. Surprisingly, they are more likely, for example, to live alone, have poorer health. So they have not specific phenotype, but it's still something that I would say it's negatively selected in the population. And this brings a very interesting ethical dilemma, because again, most people wouldn't even know that they have them. But now, if you agree to receive the results from the NIP test, actually it turns out that most people will know that they have it before they are even born. The sensitivity, it's actually, I think, about 70–80% now with the latest test. And then the question is like, what do you even do if you find an extra chromosome? I think some OBGYN will tell you it's totally fine. During the studies, you might think like it's not totally fine. And I guess it's still a little bit open whether it's recommended to report it or not, because reports have to be actionable. What are you going to do once you let the parent know that this is something that is going to affect the child? And so to conclude this section about mosaic chromosomal alterations, I show you a table here with the latest version of one test I selected. This is actually the Natera Panorama test. Sensitivity, specificity, and PPV are really high, both now for trisomy and for microdeletions, like 22q11. Other microdeletions don't have as much as a high precision. Something that doesn't show up in these kind of brochures from companies is the amount of autosomal trisomy that get missed in uncalled samples. The fact is that, as I said, for when especially when fetal DNA fraction is low, some samples will return uncalled. And those samples are more likely to exhibit presence of some of the trisomies. And you might notice here, for example, that the trisomy 18 has a sensitivity that is not 99% like the other ones here, right? Again, these are results of very large trials where even if the trial was large, the amount of positive events, you can count it almost on your fingers. And so that 94% actually was 1 out of 17 trisomy 18 missed there. And so there is a one message here is that if you compare different NIPT tests, it's going to be very hard to decide which one is best, because it's all determined on very small numbers on clinical trials. They're all very good, and the difference only happens maybe in those difficult samples with low fetal fraction. I got into the field of NIPT, and I'm going to just show you one slide for this. By actually looking at this very problem of no calls on low fetal DNA fraction NIPT. Again, as I said, I worked on mosaic chromosome alterations, and I developed a method that used the haplotypes to improve the accuracy of detection at very low cell fraction. This is a very similar problem applied to a different setting. But what we wrote a scientific report, one single trisomic case where the sample was uncalled by the commercial company. We were able to get the data from the company, and we were able to show that there are ways to improve the accuracy of the test by including what I call haplotype information or linkage information. And there are two ways to practically do that. Either you have a previous test from the mothers. So maybe this is not the first pregnancy that the mother tested for. Unfortunately, nobody has done a kind of test to show this. Like you could test if the second pregnancy has a better accuracy than the first. So another way to achieve that is if you have a really large database of tests previously run, then you can use the previous test to phase better, to infer better the haplotypes of the new test and improve the accuracy. And although I don't have any strong evidence that this is something that makes a huge difference, the latest trial from the only company that does SNP-based NIPT did mention that they use a supervised algorithm that leverage 1.6 million previous tests run, and this allows to improve the sensitivity and the PPV. And I mentioned this also because it's one of those interesting aspects that if you want to make a SNP-based NIPT nowadays, you need to have a really large database to infer haplotypes, or you won't be able to be competitive. So in the time that I've left, I want to talk about now NIPT for single gene disorder. This is a more recent area of excitement and advancement in the field. I'll start talking about like a bird's eye view of what is there to be found in single gene disorders. It's a little bit of a difficult question to ask because it depends on like what you even define as a single gene disorder. So one reference I pulled out says that maybe like 1 in 100 kids are born with a single monogenetic disorder, single gene disorder. And so that would be something like 1,000 newborns out of 100,000 births. If you look at the recessive disease aspect of it, maybe 250 out of 100,000 births will have a recessive autosomal disease. 80 will have an X-linked disease, means a mutation in the X chromosome that will affect a male. As male says, only have one X chromosome. Disclaimer, this number for recessive disease are really specific though to the US population, because every population is going to have its own recessive mutations, so its own prevalence and frequencies. If you compare that to what you could find if you care about any de novo mutation in single genes, for example, these are very small numbers. Like if you take 100,000 kids, you might find on average that 10,000 and have a de novo loss of function mutation. 60,000 will have a de novo missense mutation. It's like the majority of kids actually have a de novo missense mutations. It's probably inconsequential, but if you want to look at everything, that's where that gets you. And then here I have this very interesting plot that shows a little bit the landscape of recessive mutation in the United States, where in red you have X-linked disease, and not circled in red you have autosomal recessive disease. And here you have things that are very common, like cystic fibrosis, which is common in people of Northern European descent. HBB stands for sickle cell anemia here, which is very common in people of African descent. FMR1 is an X-linked disease, is a repeat expansion on the FMR1 gene on X chromosome. And so there are actually tests nowadays that will test for all of these genes, for mutation on all of these genes, but in the parents. And it's estimated that you might have read that it's estimated that each one of us has something like 5 recessive genes that can cause a genetic disorder. Commercial tests now exist that will scan hundreds of genes. Actually, the ACOG only recommends 5, but because it's possible, you can scan for more, and it's not very expensive. One aspect of this test is that they look for what I call the low-hanging fruit. So they look for the things that in the specific populations they test in, they are more common in frequency, because something superior is very unlikely to show up as recessive, because you need to have it in both haplotypes. But things that are more common, like cystic fibrosis mutations, that they have a non-negligible chance to show up. And so because they do that, as a fun fact, this is you don't want to use this test to condone marriages between cousins, because they will have their own rare mutation that you won't find with this test. As a fun fact, I did whole genome sequence of myself, and I found 5 recessive mutations, all validated by ClinVar, all published. Only one of those was in 1000 genome, though, so very, very private specifically. And then I found this, I noticed that if I took any one of these recessive commercial tests, I would have found 0, which I thought was very fascinating. Either I have a lot of recessive mutation, or whole genome sequence is very highly sensitive for these things in Europeans, as none would have been found otherwise. One problem, though, with this way of thinking is that you can find mutations in yourself. Mothers will be recommended to do this test when they present the clinic, usually when they're already pregnant. But that doesn't really help the pregnancy if it turns out that the parents are both positive for a recessive mutation in the same gene. The problem is that it's technically very difficult from cell-free DNA to figure out if the mother and the father are a carrier, whether the child is either not affected at all, a carrier, or with 2 copies of that mutation, which means is going to be affected by the disorder. Because again, only 10% of the DNA comes from the fetus, so you really need to distinguish between 3 different distributions. And the mother is bringing a lot of statistical noise because she's going to be heterozygous for the mutation. There is only actually one test that has a very, very careful way to quantify molecules that they devised. There's a good sensitivity in PPV, but because actually PPV is not super great, it doesn't test 400 genes. You only test the 5 genes recommended by ACOG. So cystic fibrosis, muscular atrophy, and alpha beta thalassemia. So this is still an area of development, but it's really kind of limited by the statistical sampling noise of the molecules that cell-free DNA. So there is maybe an aspect of it where if those are the molecules you're working with, at some point you're going to run out of molecules. Unless you bleed the mother out and to take all the blood, there is not going to be like an easy way to improve this, I suspect. If you think about the world of the non mutation, there is some research there. There's actually very little available in the clinic nowadays. There was a proof of concept study in 2016 where they showed that yes, you can detect mutations before birth with high sensitivity and high PPV. Well, first of all, in this case, you need to use mothers that are advanced in the pregnancy that show up with very high and usually high fetal DNA fraction. So they had 24% and 31%, which is not typical of a first trimester mother presenting in the clinic. They did an incredible amount of sequencing. This is not whole genome sequencing. This is like 10 whole genome sequence. So that even by today's standards, still probably too expensive. And you need to use a heavy amount of computational analysis to go through the whole amount of data. So at the moment, it's too expensive to perform. And then it's unclear if this would even work at the beginning of the pregnancy. There is one test that is available in the clinic. It's called Natera Vistara. They do UMI analysis. But they concentrate only on 30 genes, and most of these are genes that give phenotypes that are can be confirmed through ultrasound. And so they look at only at syndromes like Noonan skeletal disorder and some others. If you were to give a test like this to all the mothers, which is not in practice what is done, it's only given to mothers that have some weird findings in the ultrasound, 170, something like 170 days out of 100,000 would show up as positive. So this is much lower than what you would find if you were trying to look for all de novo mutations. And then there was a recent study just published last year in the New England Journal of Medicine from a collaborator, actually right here at MGH, where they said, okay, let's not do whole genome sequence, it's too much, but let's see what we can do with exome. And they showed that with exome, they can get a meaningful sensitivity and PPV for single nucleotide variants and indels. Indels is a little bit more complicated. And indels also means that STR and triplet expansions. And using a bespoke algorithm that carefully tries to distinguish noise from real mutations at low variant allele fraction, which is what you would expect since you're looking at a mixture of DNA from the mother and the fetus. They show that even at the fetal fraction as low as 9%, that they get meaningful value for sensitivity and PPV. I have a little bit of a worry that in the interest of statistics, they combine together paternity, inherited, and de novo, and they estimated the accuracy on the whole group together because they didn't have enough de novo in the exome. You only expect maybe like one de novo exome per child. But if that holds, it means that this could potentially be something that you can use in the clinic in the first trimester. But major limitations remain. If you do something like this, again, most pregnancies will receive a positive screening test. And then what do you do with that? One other problem is this works for de novo and paternity. It doesn't work for SS mutations, which have this kind of physical limit of some noise from the molecules. And then it's kind of unclear, like even if you have something and it's not a good thing, and you know maybe it's in ClinVar, it's pathogenic, but then what do you do exactly? What do you advise? It becomes something like the sex trisomy. It's like, you know, it's bad, but it's not like super bad. Like, do you really want to tell the parents? Like, how is that going to help them? What, like, which ones you should tell them? Like, and so I assume that this is going to be part of a very long conversation before we actually see these in the clinic. So to conclude, I put here a pie chart of the prevalence, average by age of the different things I talked about today. Things I want to make sure you take home as messages that, well, NIPT, especially if you're young and you don't remember this, has completely revolutionized first trimester pregnancy screening. No more scares as it used to be. Has almost completely removed the need for amniocentesis, except for the cases where actually you have positive, those 3 cases where you have positive results, has much higher sensitivity and PPV than previous standard of care, but that's important to remember, it's dependent on fetal DNA fraction. The no-call rate, which is dependent on fetal DNA fraction, is an important metric that often gets, I think, shaved under the rug. I didn't say too much, but not all NIPT tests are the same. Counting methods and the base methods are different. If you're interested more about that, come talk to me. And looking ahead, I think there's going to be a lot of innovation in single gene disorder space, especially for assessing the known mutations, but we'll see. NIPT, do know it's the fastest growing genetic test with millions of tests run every year now. I don't know the exact number, but I think it's in the several millions. And it's just bound to keep growing. And with that, I would like to thank a few people that have helped me in the journey of learning about NIPT. I wouldn't be complete if I didn't say that I was very lucky to study these things while I was in the process of expanding my family. These are my 3 little ones. Yes, we did the NIPT journey with them. Thank you so much.